Dear students, welcome to EPG Pathshala. I am Dr. Sunita Meena, scientist in Animal Biochemistry Division of National Dairy Research Institute, Karnal. Today, I am going to teach you technique of UV visible spectroscopy under paper Biochemical Techniques. Spectroscopy is the study of how light interacts with matter. We can use spectroscopy to determine the structure and functional groups in organic compounds. We will be learning about how to use UV visible spectroscopy. When light is passed through the compound, energy from the light is used to promote an electron from a bonding or non-bonding orbital into one of the empty anti-bonding orbitals. Energy of anti-bonding orbital is higher than bonding and non-bonding orbitals. Light of defined wavelength has defined energy and is different from light of different wavelength. Energy gap between bonding or non-bonding orbital and anti-bonding orbitals must coincide with the energy of light, which of course is dependent on its wavelength for an electron to get excited. Therefore, light of this defined wavelength is absorbed by matter. Energy gap between bonding or non-bonding orbital and anti-bonding orbitals will depend on which bonding or non-bonding orbital electrons are excited to which anti-bonding orbitals. If matter is exposed with varying wavelength light, it will absorb light of defined wavelength, and a plot between absorbance and wavelength is called spectrum. Science dealing with determination of wavelength, which is maximally absorbed, is called spectroscopy. UV visible spectroscopy is widely used for identification and characterization of organic compounds. In this presentation, I will be covering all aspects of UV visible spectroscopy, including electromagnetic radiation, electron excitation from one orbital to another orbital, laws governing light absorbance, instrumentation and applications of this spectroscopy. Before proceeding to UV visible spectroscopy, I would like to brief an introduction about electromagnetic radiation, which plays an important role in UV visible spectroscopy. Electromagnetic radiation or waves is composed of an electric vector and magnetic vector which oscillates in phase and 90 degrees, that is right angle to each other, and both the vectors are also right angles to the direction of propagation of wave. With the help of this diagram, we can say that direction of propagation of wave is on x-axis, electric vector is on y-axis, magnetic vector is on z-axis, and all are perpendicular to each other. Electromagnetic spectrum consists of radiations of different wavelength, each possessing different energy, frequency and intensity. High energy radiations are characterized by short wavelength and high frequency. Low energy radiations are characterized by long wavelength and low frequency. During the absorption and emission, which involves single electron transition, one quantum of the energy is absorbed or emitted respectively and is equivalent to delta E, which is dependent upon the frequency of the electromagnetic radiation. Electromagnetic radiation radiates out from a source in all the directions and includes radio waves, microwaves, infrared rays, visible light, UV light, X-rays and gamma rays depending on the source. This diagram shows the orbitals and their relative energy levels. Energy difference, which is shown in green colored arrow, falls in the range of UV and visible light energy. Ultraviolet visible spectroscopy is also known as ultraviolet visible spectrophotometry. It is based on the principle of absorbance and works in the visible and UV region of electromagnetic spectrum. This region of electromagnetic spectrum, when absorbed by the molecule, it affects the electronic transition of the molecule, and therefore transition occurs from ground state to the excited state. The interaction of electromagnetic radiation with a particular matter involves not only the properties of the radiation but also involves the structural characteristics of the matter. The electrons in a molecule are distributed at different energy levels but mainly present in the lowest energy state, which is denoted by E1, until promoted to the excited state, which is denoted by E2. The difference in the energy in terms of quantum, which is denoted by delta E, is required by the electron to reach the higher energy state, that is excited state. If this energy is derived from the electromagnetic radiation, it gives rise to the absorption spectrum, which is described as the range of wavelengths which are absorbed by the electrons of the molecule to reach the excited state. The relationship between the intensity of transmitted light and incident light depends on the path length of the absorbing medium and concentration of the absorbing solution. These factors are related in the Beer-Lambert's law. Intensity of the transmitted light, denoted by I, is always less than the intensity of the incident light, denoted by I naught. Decrease in the intensity of transmitted light is mainly because of the absorption of light by the analyte present in the solution. There is some loss of light intensity from scattering by particles present in the solution or by reflection at the interfaces. Lambert's law states that when a ray of monochromatic light of initial intensity passes through a solution in a transparent vessel, the intensity of the transmitted light is inversely proportional to the path length of the absorbing medium. In 1852, August Beer discovered another relation. Beer's law states that when a ray of monochromatic light of initial intensity passes through a solution in a transparent vessel, the intensity of transmitted light is inversely proportional to the concentration of absorbing solution. On combining both the laws, Lambert-Beer law states that when a ray of monochromatic light of initial intensity I naught passes through a solution in a transparent vessel, some of the light is absorbed so that the intensity of the transmitted light I is less than initial intensity I naught and is inversely proportional to the path length of the absorbing medium and concentration of the absorbing solution. Transmittance, denoted by capital T, is the ratio of intensity of the transmitted light and intensity of the incident light. 100% transmittance represents a solution which does not absorb anything and 100% photons reaches the detector. 60% transmittance represents a solution which absorbs 40% of the photons and 60% of the photons reaches the detector. 0% transmittance represents a solution which does not transmits but absorbs all the photons. Absorbance, denoted by capital A, is the logarithm of reciprocal of transmittance. Absorbance is equal to 0, represents no photons are absorbed by the solution, means no analyte is present. Absorbance is equal to 1, represents 90% of the photons absorbed by the analyte present in the solution and only 10% of the photons reaches the detector. Absorbance equal to 2 represent 99% of the photons are absorbed by the analyte present in the solution and only 1% of the photons reaches the detector. By taking the log on both the sides of the equation, absorbance of the solution is the product of extinction coefficient, concentration of the solution and path length of the cuvette through which the light travels. Molar extinction coefficient, also known as molar absorbance coefficient or molar absorptivity, is equal to absorbance when the concentration of the solution is 1 mole per liter and the path length of the cuvette is 1 centimeter. Spectrophotometer covers the range of wavelengths in both the UV and visible region. Materials used in the spectrophotometer depends upon the wavelength used. Tungsten filament lamps are used in the visible region and hydrogen and deuterium lamps are used in the UV range. In UV visible spectrophotometer, both the lamps are switched on, covering the visible as well as the UV range, and it depends on the user which lamp to be used for which purpose. For this, a mechanical switch is available for directing the light along the optical axis. Monochromatic light refers to the light of single color or single wavelength or narrow band of wavelengths. Monochromator is an optical instrument which selects the light of single or narrow range of wavelengths from wide range of wavelengths emitted from a light source. Wavelength is selected by the monochromator such as prisms or gratings. Prism separates the beam of light into its components by the phenomenon of refraction or dispersion, whereas gratings do this by the phenomenon of diffraction. The resolution is greater from gratings than from the prisms. Optical slit width affects the bandwidth. Light from a source is emitted in all directions. Slit allows only a portion of light to reach the monochromator, and the selected light is again allowed to pass through the slit. The reproducibility of the absorbance values increases as the slit width decreases. With the decrease in slit width, sensitivity also decreases as less light reaches to the detector. Sample for the detection should be placed in a transparent cuvette, mostly rectangular in shape and commonly have internal width 1 cm. Small test tubes are also used in some instruments. Cuvettes are made of silica for the detection in the visible range. Borosilicate glass is used in visible region as well as in near UV region. Fused silica and quartz cuvette are required for the UV range, but also covers the visible and near infrared region. Chromophore is the part or moiety of the molecule which is responsible for imparting color. This phenomenon occurs when a molecule absorbs certain wavelength in the visible range and promoted to the excited state. Also, the absorption can occur in the UV range. Biological molecules also cause conformation change when exposed to light. Examples include molecules or compounds that have unpaired electrons or unsaturated bonds which can absorb the radiation, such as alkenes, ketones, aldehydes, phenyl and other aromatic species. A spectrophotometer may be single beam or double beam. In single beam spectrophotometer, beam of light passes through the cuvette present in the sample cell. Therefore, sample cell is first used to zero the instrument by using blank or reference solution in the cuvette, and later by placing the sample cuvette in the same place, and the absorbance is recorded. This is to minus the blank absorbance readings from the sample absorbance readings. In double beam spectrophotometer, incident light is split into two beams by half mirror. One beam passes through the reference cuvette and the other beam passes through the sample cuvette. The transmitted light from both the cuvettes then reaches the detectors. So far we have discussed the UV and visible range of electromagnetic radiations and its interaction with the matter. UV visible spectroscopy is based on absorbance phenomena, where the electrons absorbs these radiation and moves from ground state to excited state. This has been explained by Lambert-Beer law, that is, the absorbance is dependent on path length of absorbing medium and concentration of absorbing solution. We have also discussed the instrumentation of UV visible spectroscopy, such as light source, which can be tungsten filament lamp for visible region and hydrogen or deuterium lamp for UV region. Monochromators, which selects the light of single or narrow wavelength. Prisms and gratings can be used for this purpose. Slit is also important as it allows only a small portion of light to pass through it and reproducibility increases as slit width decreases. Cuvettes are transparent and these should not absorb in UV visible range. And lastly, we have also discussed the difference between single beam and double beam spectrophotometer. By now, you must be clear that lambda max is the property of chromophore, which is the part of molecule. UV visible spectrophotometer has vast applications. To be concise, major applications include qualitative and quantitative analysis, different spectra and binding spectra. UV visible spectrophotometry can be used for the identification of molecules, compounds in its pure state as well as in the biological mixtures. Examples include proteins, nucleic acids, chlorophylls, cytochromes. This technique also gives the structural details of proteins and the intermediates arising in a biological system. It is also used in the analysis of transition metal ions as they absorb in the visible range, because of which the d electrons are excited to the higher energy state and gives color to the metal ion solutions. A plot of absorbance or %T against the wavelength can be obtained. However, getting a plot of absorbance against wavelength is a common practice. With the help of the standard curve, quantity of unknown molecule in a sample can be determined. Many colorimetric assays are used in the visible range for the easy detection of the molecule. A molecule which is to be detected may be colored or may be chemically modified to produce colored product. Proteins and nucleic acids are also measured in UV range. Proteins are measured at 280 nanometer and nucleic acids are measured at 260 nanometer. For pure DNA, absorbance at 260 by 280 nanometer is approximately 1.8. If the ratio is more than 1.8, it indicates the RNA contamination, and if the ratio is less than 1.8, it indicates the protein contamination. For pure RNA, absorbance at 260 by 280 nanometer is approximately 2, and for pure protein, absorbance at 280 by 260 nanometer is approximately 2. If the value of molar extinction coefficient is known, it is possible to calculate concentrations directly from absorbance readings at specific wavelength. For pure proteins of known coefficient, the concentration can be calculated by dividing the absorbance at 280 nanometer by the extinction coefficient. The concentration of the proteins with possible nucleic acid contamination can be calculated by the formula 1.55 into absorbance at 280 nanometer minus 0.76 into absorbance at 260 nanometer. Although proteins and nucleic acids can be measured in UV range, there is an additional requirement of corrections for the interfering substances. A difference spectrum is the difference of two absorption spectra. There are two methods of obtaining the difference spectra. First, by subtraction of one absolute spectrum from another. Second, by placing one test compound in the reference cell containing the compound in reduced state and the other in the test cuvette containing the same compound in oxidized state. Different spectra may have negative values. The absorption maxima and minima is different from the individual spectra of reduced and oxidized compound. Isobestic points are also observed in the different spectra where the absorbance values in individual spectra are same. The main advantage of different spectra is that it enables the detection of small changes in absorbance occurring in a biological system with a high background absorbance. Examples of such system is the mitochondria and chloroplast, where it is possible to detect the changes in the oxidation states of its components. Here is one example. The spectrum of ubiquinone, which is oxidized, and ubiquinol, which is reduced, is presented here on left hand side figure, and the right hand side figure shows the different spectra. Different spectra is obtained by subtracting the absorbance values of ubiquinone from ubiquinol. Binding spectra is also known as substrate binding spectra. As the name suggests, this is used to study the level of interaction between an enzyme and its specific substrate. Binding of a substrate or drug to enzymes can be detected by changes in the absorbance or changes in the lambda max. Upon binding, there may be increase in absorbance, which is known as hyperchromic shift, or decrease in absorbance, which is known as hypochromic shift. Binding of substrate also causes shifts in lambda max. Conjugation of double bonds lowers the energy required for the electronic transition. Therefore, the chromophore absorbs at longer wavelength. Increase in lambda max is known as bathochromic shift, that is red shift. A decrease in conjugation increases the demand for higher energy wavelength to be absorbed by the chromophore, causing a decrease in lambda max, which is known as hypsochromic shift, that is blue shift. Dear students, if I summarize what has been covered in today's presentation, I can say UV visible spectroscopy is very powerful spectroscopic method for identification and quantification of analytes. Visible range is used for colored solution, while UV range is used when molecules have characteristic bonds. The glass cuvettes are used for visible range, and quartz cuvettes can be used for both the UV and visible range of electromagnetic radiation. Concentration of analyte can be calculated from its absorbance and the molar extinction coefficient. Double beam spectrophotometer is preferred over single beam spectrophotometer, as it reduces the effort of subtracting the blank reading from the sample reading. The technique is routinely used for assaying analytes. It has wide applications beyond what is being discussed. The focus of this presentation was to make you understand the basic principle, instrumentation, and applications of UV visible spectroscopy. Thank you for searching this video on EPG Pathshala.